So I'm Dylan. I'm a postdoc here at PNNL, and today I'll quickly kind of go over a tool we've been working on for the automated elucidation of double bond positions from Oz ID data we call LipidOz. So very briefly, luckily for me, Professor Fernandez this morning kind of spoke about the importance of annotation in across omics technologies, but particularly the case in lipidomics, where there's kind of this complicated way of annotating lipids, right? So depending on your instrumental conditions, your experimental design, you can identify lipids at sort of varying degrees of structural detail, right? So if you think about a lipid, it's kind of composed of this head group, and that corresponds to the lipid class. It typically will have, one or more fatty acid substituents attached to it. And those can vary in terms of, the length of the carbon chain and presence and position of these double bonds or unsaturations. And so ultimately, ideally, you would know not only the lipid class, but the specific orientation of these fatty acids, as well as the position of double bonds in the lipid. That's kind of functionally about the highest degree of structural detail that you can kind of annotate a lipid with, and that has important biological implications. So that's sort of the motivation for wanting to sort of reach this high level of annotation. And so there are a number of ways of kind of experimentally getting at this information. One way that we use is called ozone-induced dissociation, or OZ-ID. And so the kind of main functional premise here is that in the lipid acyl chains, you have, double bonds. Those can interact with ozone, forming this unstable species, which breaks apart in a predictable way into these pairs of fragments. We call them the aldehyde and the Craigie. And these fragment ions differ in mass by 16 Daltons, and importantly, their masses are diagnostic of the position of this double bond in the lipid. So to make that a little more tangible, have this kind of sort of prototypical lipid here, and you can see it's got these 3 double bonds. When the ozone reacts with each of these, you get these pairs of masses, right, with, again, diagnostic masses, or these pairs of fragments with diagnostic masses that'll tell you where these double bonds are. And so that sort of manifests in, our prototypical mass spectrum here. And so the trick with this technique, though, is that the data analysis process is pretty complicated, as you might imagine. So I'll just very quickly kind of run through what that looks like for one target, just to sort of highlight this fact. So under this experimental design, we start off by extracting an extracted ion chromatogram. For the precursor species, we can, select in a region of this to extract out the mass spectrum for that precursor. And then using the formula of the precursor, we can predict what the theoretical isotopic distribution should be. And that's kind of these red dashed lines compared to, what we observe experimentally. And this is kind of an important quality indicator, or sort of sanity check, if you will, or validation of the precursor identity. So we can then, knowing, some basic information about the precursor, but not knowing the double bond position, we can kind of predict for all of the possible double bond positions in this lipid, we can predict those diagnostic fragment masses. And the data processing workflow for each of those is kind of similar to what we did with the precursor. So here I've got kind of 2 examples of putative fragments, one corresponding to, let's say, the N-6 position, that's 6 in from the tail, or another corresponding to N-9. And so like with the precursor, we extract the XIC. We can select kind of the tip of the XIC and extract mass spectrum, compare that to the theory based on the molecular formula of the fragment. And the 2 kind of critical indicators, or sort of metrics we use for assigning ultimately a double bond position from this data are, number 1, the agreement between this XIC of the fragment, the putative fragment, and the precursor, because if it's produced from the precursor, they should have similar chromatographic profiles. And number two, the agreement between kind of the theoretical and observed isotopic distributions. So in this case, it's pretty clear, this N-9 putative fragment, matches well in terms of its XIC, and, it has really great agreement with the theoretical isotopic distribution. So we would say, this is the likely annotation for this species. And the main point here is you can imagine the many number of, the complex data processing, right? We have to go in, extract XICs, extract MS1 spectra. There's a lot of steps, and there's also a ton of repetition, right? So for one, just this species alone, there are like 16 possible double bond positions you have to check for pairs of fragments for. And so obviously doing this manually is just completely impractical. So we set out to kind of automate this process, and we did so using Python, and this culminated the development of a GUI application that kind of goes through, or before I step through, that kind of goes through this whole process in a pretty automated fashion with some minimal kind of parameter setting at the beginning. And so we have both a kind of Python API for being able to kind of monkey around with this and do kind of custom workflows to your needs. But we also kind of package this into a standalone GUI application, which I'll just quickly run through a little demo of how that looks using it. So it starts up with this screen. It's kind of our setup screen. And there's sort of two sections, right? This upper section is for setting up kind of data processing run. The lower section is, for viewing results that you've already processed before. But basically, the inputs are pretty straightforward. We select, the data file. And again, we're using this EMSL format developed by IVET, which is really convenient for a number of reasons, but I won't spend too much time on that right now. And then a target list of lipids for which you want to annotate the double bond positions. And then the next set of inputs are basically just parameters that control how the data is extracted and processed. And this includes basically some allowance for having deuterium labels. So deuterium labeled lipid standards are pretty common. So we have the ability to kind of accommodate those. But yeah, when you're all set up to go, you can go through and process the data. That'll pop up this processing screen, and pretty straightforward. It just, this process takes a while. Like I said, there's a lot of steps and a lot of repetition. And if you have a particularly large target list, it'll take a while to chug through all of that data. But so this just gives some info to let you know that it's not just hanging, that it's doing stuff. But eventually, when that's done, you'll see this, view results button is no longer grayed out. And we can hit that and proceed on to what is probably the most, is kind of the main part of the GUI application, is the sort of interactive data viewing. And so this GUI is organized into a couple of kind of panes. So on the left side here, this panel is sort of the lipid target list. It's kind of a tree view menu. And then the panel to the right here displays all of the extracted and processed data. So the top two plots are for the precursor, and then the bottom two, or the bottom four plots, are for the two, putative fragments for a given double bond position. And so if we go ahead and select out, kind of expand this menu down, I'll also note, we have basically, we can accommodate, looking for multiple adducts or ionization states for a single lipid species, in addition to multiple retention times. But here, an individual sort of set of results is defined by double bond index, just how many double bonds on the chain is it. This is basically comes into play for polyunsaturated fatty acids. In this example, it's just monounsaturated, so there's just one. But the main part here is this list of putative double bond positions, and it's sorted in decreasing order of kind of confidence. And this confidence is based on, again, those two mass spectral and chromatographic kind of components that we use as criteria for assigning double bonds. So if we select, this most confident double bond position here, we see, as expected, if we look at the precursor, its isotopic distribution looks good. We look at this pair of fragments, both of them have XICs that match the precursor, fair enough. fairly well enough, and then their isotopic distributions match what we'd expect from their formulas. So kind of makes sense that that one looks great. If instead we take a look at one of these more horrible ones, we don't even have any data. We aren't able to extract like a fittable XIC for the Craigie ion or Craigie fragment, and then this fragment has a retention time that's clearly not aligned with the precursor, and it's got some pretty bad mismatch between the observed and theoretical isotopic distribution. So it really makes sense why that one's at the bottom of the list. Now, if we go and take a peek at one of these, a polyunsaturated fatty acid containing one target species, we can see there's a lot more data to look through now, right? Because for each of the 4 double bonds in this fatty acid, there are a set of putative double bond positions. And, as it stands right now, it's kind of hard to really deal with this data. So we have basically functionality in here where you can go through and basically just delete results that are total garbage. And so, oh yeah, as I've kind of highlighted here, we could kind of get rid of all of these. And now we have, for every double bond index, we have just a single double bond position left that has, good scores. We can kind of like go through all of those and we can see that, okay, for, index 1, looks like double bond position 6. The next double bond looks like it's at 9. The next double bond looks like it's at 12, and the final at 15. That turns out with 24, that's arachidonic acid. That makes sense for the sample. And yeah, finally, we can save the results. That just saves it in a binary format that you can load again later from the setup window, like I alluded to earlier. And then finally export results. And this exports to basically this whole tab, this data. It sort of explodes that into one big Excel spreadsheet. And that spreadsheet also has it has a sheet in it for kind of metadata. I guess the text doesn't render super well at this size, but basically these are all the parameters that you would have entered in the setup window anyway. So if you need to go back and rerun something or understand, what parameters did I use when I ran this, that's where that information is. And then the targets sheet has basically all of the data that you were able to kind of interactively view in the GUI, but in a extended tabular format. So like the first set of columns here are a bunch of information mostly about the precursors, and then the next set of columns are about the aldehyde and then Craigie fragments for different putative double bond positions. So if you were to like kind of imagine, zoom way out and squint at this whole table, we have our precursor information, then aldehyde and Craigie. And so like you'd look for lines that say have, high scores. So kind of this like blue color coding kind of across the board where the precursor looks good and, the pair of fragments looks good. Okay, so that was sort of, so that analysis is kind of a more standard or traditional, basically automating the steps that an expert would take as they sit and work through the data. And it's got all these like complex stepwise sort of considering each piece of information in turn. But if you think about this problem in a slightly different way, there's kind of an interesting way of formulating it, and that is when it comes down to evaluating different double bond positions, the task kind of decomposes into a pretty simple comparison. So for one double bond position, we're comparing LCMS profiles, that is two-dimensional data that consists of a mass spectral, kind of in the X dimension here, and retention time components in the Y dimension. Similar to what actually Yvette was showing earlier, right? So we're comparing these profiles for the precursor in a pair of putative fragments that are diagnostic for one position. And so when we evaluate different positions, we're basically just comparing different sets of these 3 LCMS profiles. But these LCMS profiles, like Yvette said, look kind of like images. And so we could kind of construct these, consider each of the, the precursor and the 2 fragments each as individual channels in one RGB image. So now the task really kind of boils down to look at RGB images and decide, what the double bond position is based on, what pattern looks most correct. And so in this case, under this formulation, this is obviously like a perfect application for deep learning. So I'll just like super fast go over the kind of the workflow that we use for actually training a deep learning model that can do this type of analysis. So it starts off with extracting raw data. Again, these are those, sets of precursor and fragment pair LCMS profiles. As you can see here, kind of the data that you initially get is sort of sparse kind of scan data. So then we apply some binning and normalization to get those into kind of a uniform size and shape that'll play more nicely with the downstream machine learning architectures. And we also do some data augmentation by kind of jiggling around the retention time dimension. And that's to give us a lot more training examples and hopefully make the model a little more robust to slight shifts in retention time. We then split the data into training and validation sets. This is pretty straightforward. I just wanted to mention, we used 3 different sets of kind of standards, in training our model here. And, 2 of these are like deuterium labeled authentic standards that we know the exact positions. This 3rd one here is kind of a standard lipid extract that we've run a lot and is well characterized. So we kind of know what to expect from it. And then finally, I don't think I need to belabor the general machine learning workflow too much, but I will mention that, especially because we are able to formulate our data as RGB images, we're able to use this off-the-shelf deep learning architecture that is already trained for image recognition tasks. So we use that as our starting point and then retrain it for the classification task of assigning lipid double bond positions from the SAWS ID data. And the real take home here that I want to hammer is that, this sort of deep learning workflow isn't like a wholesale replacement for the more traditional automation that I was talking about earlier. In fact, without that traditional automation, we would never be able to curate a sufficiently large data set to actually do this deep learning. So, these 2 approaches are really pretty synergistic. And so I'll just quickly run through, I mentioned before with the kind of traditional automation workflow, we have both sort of a Python GUI that enables some kind of more customization in terms of, your workflow needs, as well as that GUI application. And likewise with this deep learning workflow, we have also the Python API, but we don't have a kind of bundled GUI application for this. But I wanted to just highlight, how easy it is to kind of tackle this yourself. Let's say you have a data set that you'd like to train a model to, be specific to, or you'd like to, enhance what's already there. Basically, I just wanted to highlight what we have in the API for doing that. So kind of for the first step, extracting raw data, we have this function collect pre-ML data set. That is kind of a unmodified pre-machine learning data samples. And basically, it just takes similar inputs to the traditional workflow. We have our data file, we have a target list of lipids, some other parameters that control how the data is extracted. But ultimately, we get this pre-ML data set, which basically just consists of some metadata. And then for all of your lipid targets and potential double bond positions, you have your precursor aldehyde and Craigie data. So that's the precursor and then the pair of fragments, as kind of sparse array data. And the point here is this function really just takes care of all of the actual like chemistry calculations and determining all the possible double bond positions and the corresponding masses and all that for you, and just gets you to some data that's ready to be processed, which is kind of the next step, right? So taking this sparse data and putting it into a nice clean form that can be fed into the deep learning pipeline. And so that's just done by this, convert multi pre-labeled data sets. And so we start with, potentially multiple of those pre-machine learning data files. So those data sets that we produced before, and then some basically target files that tell you, the authentic, or that tell this the authentic double bond position labels. So this is like a supervised method, right? And ultimately, this just goes through, it does, the retention time sampling, augmentation if you want it, normalization, binning, all that stuff. And ultimately, you get this nice array data split out by true and false annotation. And, they're all in these like nice, clean kind of RGB images, right? So 3 channels, 24 by 400 pixels. So these are kind of now ready to go into the machine learning pipeline. And so we also have kind of this next function here. Basically just takes that kind of labeled true and false data that has already been processed and ready to go, and splits that into training and validation sets. And it also, so we're using PyTorch as our kind of deep learning framework in this. And so that requires, it carries certain requirements with regard to, how you can hand data over to the model. So this takes care of setting that all up and gives you these data loaders that the model can actually use to load data and do the training. And then lastly, we have the training step. And like I said, we used ResNet 18, but this interface is actually, doesn't really matter what model architecture you use. So it's kind of a base class, and it has this train function, or train method rather. And that just takes those data loaders and some parameters for how you want to train it, including, what kind of loss function, what optimizer, how to schedule the learning rate, how long to train it, that sort of thing. And so, yeah, you could, I guess, define your own DNN architecture if you'd like, or use an off-the-shelf one like we did with ResNet 18. That's all kind of up to you. Yeah, so, and then the last point here I want to make is, once you have a trained deep learning model, the process of going from raw data to identified or annotated double bond positions is pretty much a couple of completely automated steps. So we've gone from, a horrific manual data processing to something that's really streamlined. And that's kind of the power of this deep learning approach. And just to prove to you that, this actually works. I have here the confusion matrices from the 3 individual sets of data that I talked about earlier that we trained this model with. And you can see that in this particular case, we actually managed to get, no mispredicted values or wrong predictions. So we were able to get, 100% accuracy across these, which was kind of surprising. But yeah, I think pretty well showcases how nice of an approach this is. So yeah, just to wrap up really fast for sake of time, this application, we're kind of in the final stages of being ready to submit our manuscript on this, and we're going to have all of the API, both the kind of traditional automation and deep learning portions of it. We have a nice Python API with documentation. It's all going to be open sourced, and we'll also have that kind of bundled GUI application for the kind of traditional automation flow. And this package here, or this whole suite, is built on top of ultimately the EMSL mass spec, open mass spec data format. Again, developed by IVET. This is really awesome, and this is, I think, a good example of when you remove kind of the restrictions that are imposed by relying on certain vendor formats and things like that. It makes it really easy to build application stacks on top of that. And looking forward, we've got a few more things that we're working on in the lipidomics space and beyond. But for sake of time, I'll just quickly like to shout out kind of the core LipidOz development team: Chiyun, Ivet, and Junyoung Lee, and then the rest of the kind of PNL folks who do all the data generation and stuff like that, and external collaborators, funding, and thank you all and the organizers, and take any questions.
All right. Thank you so much. That was a great presentation. My question will go about the data input, because I think it's where everything, everything works. If you don't have a good input, you don't have a good input data. Okay, so how do you define your input data? Let's say I have no idea about your MS data, and then I just want to use my own data with your program, right? How am I going to define my input data? How will my input data look?
Gotcha. So in this particular, for this particular software, we are sort of targeting a very specialized data analysis that comes from like a pretty specific experimental design. And apologies for time, I didn't really go over kind of the experimental side of things, but it does involve kind of a modified like ion mobility instrument that can do this ozonolysis. But generally speaking, maybe, outside of this particular application, using, if you're able to put your data into kind of a format that's, more, that's like an open mass spec format, there are, actually, even with some of the work we've done here, there are tools that you can quickly develop tools that are sort of tailored to your own workflows pretty easily. Yeah. So once it's sort of in a format that's easier to kind of program with, the barrier is a lot lower. So I would say that's kind of like the first point is, dealing with the mass spec data format issue.
Thank you. And also, ask one last question. Yeah. Okay. So also, you didn't mention too much about the. How good is, how good is your model can also depend on which kind of feature you're looking for, right?
Yes.
Yeah. So then, what feature or what other parameter are you actually using?
That's an excellent question. And I probably could have made it a little more clear. So I'm specifically using a deep learning architecture. So that is like a deep neural net. And the benefit, thanks to Ivet for spending some time to explain that, though, the benefit is that you don't necessarily need to do explicit feature engineering, right? So in this case, we just feed directly these RGB images, and we let the model decide, what it is in any of these channels that it wants to really look at and key in on and, activate strongly from. So that's the benefit of these deep neural network architectures. And even easier, I guess the last point I'll add is the fact that we were using this off-the-shelf ResNet 18. It's already been trained on insanely huge sets of like image data. So it already has some very complex, it's already learned some very complex kind of image patterns to activate on. And so we just took that and trained a little bit, a few layers at the bottom, more or less, just specifically to our task. Mm-hmm.
Do you have any other questions? Caroline? Thank you, Dylan. And thank you, everyone, for attending this session. Feel free to contact us, and if you have any questions, if you are interested in lipids, yes, you can put a proposal and use our tools. And as Professor Fernandez was saying, that there are different levels to characterize the lipids, and they are very important also for all living systems, also for bacteria. And actually, Dylan has processed some data also that will go in his publication showing how useful it is. So using this technique, you can go into more details to understand and characterize the structure of these lipids. Thank you.